So good evening everyone. Today is our week 9 session. So in this session we'll be looking into the molecular visualization tools, in which we'll see the history of different molecular visualization of biological macromolecules. We'll also see that what are the different structure related files with which we can store the information of atomic coordinates and other stuff such as PDB, MMSIF, MTZ, etc. Then we'll also look into COOT and how we can visualize the protein tertiary structure and other things using PyMOL. And then we'll go into the details of PyMOL, that what are the different things that we can do with the help of PyMOL. Later on we'll practice few MCQs, we'll look into the current literature and I'll take your doubts. So if we talk about the molecular visualizations, then the question arises that why it is so important. So if we have a molecular visualization, then it helps us to understand the abstract scientific concepts which are not visible to our naked eye. So due to which the computer modeling is a tool which can provide us the window to the unseen, which can revolutionize the scientific research. So due to which we require the molecular visualizations, because there are multiple amino acids and all those things which interact with one another and perform the chemical reactions. So we cannot see all those things with the naked eye. So that's why, to understand all those interactions and how the protein molecule behave in which environment, so we need to understand that what is their molecular structure. So due to which the molecular visualization becomes very important. Then we'll talk about what are the historical methods. So before computer visualization, which we see nowadays, previously the molecular structures, they were represented using the physical models such as metal wires, rods and spheres. So as you can see in these figures, so previously those structures used to be represented by the help of these metal wires and these balls. So when the computer visualization was not available, so in 1958, Kendrew was a scientist. Along with his colleagues, he used brass models to visualize the myoglobin structure. So its scale was 5 cm per angstrom, which was supported by these vertical rods. So this is a brass model for the myoglobin given by Kendrew. Then with the help of computational methods and advancement in computational technology, there was development of hardwares and softwares which has enabled 3D visualization and manipulation of these molecular structures. So now we can visualize these molecules in 3-dimensional using computational tools. Instead of using the physical models to see the protein molecule, we can see them in the computer screen. And these graphics, they also help us to analyze and compare protein structures to determine their functions. So what are the applications? So it assists scientists and biomedical experts in studying, understanding and engineering biological macromolecules, and it also helps in protein structure function analysis and molecular engineering. So how it can help in protein structure and function? Because protein structure, it depends that how the protein is folded, what amino acids are exposed, what molecules are buried. So depending on those molecular properties, the protein can function in a certain way. So due to which, if we understand the structure, then we can also understand its function. So that's why this molecular visualization is very important. Now if we talk about how it began and where we are right now in terms of molecular visualization, so we can see in 1962 these two scientists, they won Nobel Prize. So one was Perutz and another was Kendrew. So they got Nobel Prize in chemistry in 1962 jointly for their studies of structure of globular proteins. So previously we have seen that how Kendrew model of myoglobin helped to physically see that how a protein structure looks like. So they got Nobel Prize in 1962. And if we talk about current time, so in 2024, these three scientists, they have got Nobel Prize in chemistry and they have worked on the so if we talk about David Baker, so he worked on computational protein design, whereas these other two scientists, Demis Hassabis and John Jumper, they worked on protein structure prediction. So I hope you are aware of the name AlphaFold. So they have given this AlphaFold tool with which we can predict the structure of unknown protein structures, the sequences which are not previously solved by X-ray crystallography, NMR. So those structures can now be predicted with the help of AlphaFold. So these are the advancements which have taken place, and these are this AlphaFold, it works on the computational representation of protein structures. So in later slides we'll see that how these things have facilitated. But before that, we'll firstly talk about the earlier methods of molecular visualizations. So starting with the Richard box, which is also known as Fred's folly. So it was invented by Fred Richards in 1968, and this is also known as optical comparator. So why it is called optical? Because it takes the help of the mirrors. So in this case, they tend to use the half silvered mirrors to superimpose the electron density maps on the brass protein models. So in this case, this is a half silvered mirror, and these are two electron density maps. So with the help of this half silvered mirror, they used to superimpose them onto a brass model. So while looking at that brass model, they used to visualize the electron density map. So in this way, the scale was reduced over time, starting with 2.5 centimeters per angstrom to 1 centimeter per angstrom for larger molecules. Yeah, and this is what it actually looks like in 3D. And then came physical ball and spoke model. So it was developed by Kendrew, who solved the structure of myoglobin, and it represented the molecular structure using the balls as atoms and spokes as bonds. So this is how the ball and spoke model looks like. Then comes Byron's bender. So in this case, it was invented by Byron Rubin in 1970s. So by the help of this bender, they tend to bend the wires to show the protein backbones. So these backbone wire models, they are highly manipulable and portable, because with the help of this bender, we can easily bend it, and we can easily change it, we can easily manipulate it. So that's why this model was easy to carry. So that's why it was much preferred. Then came molecular sculptures. So there was a sculptor named Bethesda Grossman who created the laser crystals by rendering the proteins as tiny laser-induced fractures within the glass blocks. So these are the glass blocks, and with the help of laser, they used to fracture them from inside to represent the protein structure. And then there were another artists also who made large protein sculptures in the metals or woods. So with the help of metals and wood, different other sculptures are also there who use them and to represent the protein three-dimensional representations. Then rapid prototyping. So in this case, it initiated in 1990s by Michael Bailey and Tim Herman. So they used rapid prototyping, which is also known as 3D printing. So in this case, they created 3D physical protein models with the help of support wax or willed wax. So they use the mold printing. So they have molds with which they tend to print it, followed by dissolving the support material. So in mold printing, what they used to do, they have a support wax and a willed wax. So they have a mold in which they put this wax, and then later on they used to get the structure with which they can easily represent that which of the residues are towards the surface or the periphery, and which residues are lying inside of the protein molecule. So this also was the method which was used for the visualization. So this one is the earlier method. So okay, let's see. Yeah, so these were the earlier methods. Then came computer representations. So before this, we have seen these all were the physical methods, but as soon as the computer technology advanced, so we have now computer representations. So the earliest computer representation was in 1960s and 1970s. So in 1964, there was a scientist named Cyrus Leventhal and his colleagues at MIT. So they developed a system to display rotating wireframe representations of macromolecular structures on an oscilloscope. So this is how it used to look like with the help of oscilloscope. So this is the rotating wireframe representation, and this was a computer representation. Then came OTEP, which is a Oak-Ridge Thermal Ellipsoid Plot Program. So it was released in 1965 by Carroll K. Johnson, who produced the stereoscopic molecules and crystal structure drawings. So these drawings, they are known as the OTEP representations. So they became widely used by the crystallographers for illustrating molecular structures in publications, presentations. So in 1966, Johnson used this OTEP to create stereoscopic drawings of myoglobin, vitamin B12, coenzyme and polyl-alanine. So these are few of the molecules for which he designed these drawings. and this is the Carol K. Johnson who developed this method. So the key advantage was that the computer tracked atomic coordinates, eliminating the need for manual measurements as required in Kenryu models. So before this, all the physical models which we have seen so far, so there were problems that we need to track the atomic coordinates manually. So we need to make sure that all those things need to be taken care of. But in case of computer representations, we do not have to worry about the atomic coordinates. So we do not have to perform manual measurements and convert the angstrom measurements into centimeters or all those things. So they have simplified the computer representations. So they have simplified that how the protein can be visualized. Then came MOL script. So it is a program for generating molecular graphics from 3D coordinates, and it is primarily used for the case of proteins. And the users provide an input script defining the coordinate file, objects and rendering parameters. So this is how the protein architecture looks like in MOL script. So in this case, they properly shows that where are the alpha helices present, where are the beta sheets present. So in this case, the periphery is abundant of the alpha helices, whereas in the middle there are beta sheets, so it looks like a alpha beta barrel. Whereas in this case, these all are the alpha helices, so they are the alpha helix bundle, and this is the ID of the MOL script. Then this is the solenoid in which these beta sheets, they are represented in the form of the solenoid. Whereas in this case, these are the alpha horseshoe, because this alpha helices, they combine to form a structure like a horseshoe. So in this way, with the help of MOL script, we can generate this molecular graphic structures. Then came persistence of vision ray visualization. So this is also a cross-platform which is based on the ray tracing method, which creates the images from the text-based scene descriptions. So ray tracing simulates the light from the camera to render realistic scenes. So with the help of this POV ray visualization, we can see the molecules from different directions, and then we can superimpose them to see that how that particular molecule will look in a three-dimensional space. So you can say that this is a kind of taking 2D pictures from different directions and then combining them or merging them to see that how it will look in a three-dimensional space. So this is known as POV ray visualization. Then came TAMS, which is known as teaching aids for macromolecular structures, so which create colored atlases of computer generated molecular images. So previously, the generation of colored atlases by using the colored computer generated molecular images, it was quite expensive. So a cost effective solution which emerged with a simple cardboard stereo viewer for 35mm slides in 2x2 inch mounts. So this was the TAMS. With the help of this particular tool, we can visualize the macromolecular structures which appear like they are in the 3D form. So it could be folded flat for easy storage in the cover pocket of the TAMS 3-ring binder. So this was easy to store and easy to carry. So with the help of this TAMS, what happened is that, for example, it has 2 holes, and a person can watch with his 2 eyes from these 2 holes. So what it will do, if we have 2 different 2D structure of a protein, then if we see with these 2 stereo viewers, then it appears these 2 will overlap on each other and it will appear as a 3-dimensional structure. So for example, in this case, if we watch these 2 trees with only one lens of this particular stereo viewer, then we will not be able to capture how they are present and at what difference both the trees are present. We won't be able to clearly capture them. But if we look from both the holes of the stereo viewer, then we can clearly figure out, and we will be able to see them as a separate figures, and we'll be able to generate the figure in our mind how they are present in 3-dimensional space. So this is the concept of stereo viewer in TAMS, that if we have 2 different images present next to each other, so while looking at these 2 holes, we can combine them, we can overlap them, and we'll be able to see them in the 3-dimensional form, in which we'll be able to know that which of the parts in that protein, which of the residues are surface exposed and which parts are buried. So it gives us kind of a depth information of a protein. Then there are different computer systems such as Evans and Sutherland computers in 1980s. So these are the high-end computers, and they were about this price range, which were used for visualizing the electron density maps. which allowed manual fitting of amino acid sequences into the map and featured real-time rotatable wireframe rendering of amino acid chains. So with the help of these high-end computers, we can visualize the electron density maps, and by looking at those electron density maps, because we know that what is the electron density, so we can fit manually the amino acid chains into it. So we'll be able to know which residue can fit in which of the electron density position. So in this case, as they have fit tyrosine here, so it clearly, if we keep the tyrosine residue here, then it will generate the electron density map of this sort. So this is the manually placing the or manually fitting the amino acid sequence over the electron density map. Then David and Jane Richardson's Kind Mages. So Kind Mages in 1992. So they have pioneered the molecular structure visualizations through interactive graphics. So these Kind Mages stands for kinetic images, which emphasized selective representation over neutral depiction. Integrated author's perspective highlighting the specific structural aspects. So in this case, these are the kinetic images which keep on changing, and they used to check that whether there are some Ramachandran outliers or either there are steric clashes or bad puckers. So in this, because we have now kinetic images, so we can make sure that the image which is generated in the end, it should have least steric clashes and it should lie on the Ramachandran favorable regions. So all those things are checked, that whether this model or the protein structure is favorable or not. So in this way, multiple images are generated, and the one which supports or which favors all the prerequisites for a proper protein structure or stable protein structure is the one which we consider. Then there are other scientists, so such as Roger Seil's RasMol. So this is also another tool in 1993. So the purpose was to design for a self-directed neutral exploration of molecular structures, because in the Kind Mages what we are doing is we are kind of biasing it on the basis of Ramachandran outliers and steric clashes. So we are kind of biasing it. We are considering different conditions. We decides that what should be the value to consider a protein to be, or to consider a model to be valid or not. So we need to mention those threshold values in this Kind Mages. So we are kind of biasing it, because the person who is deciding those threshold value is the one who has the control. So these values can vary because it is in the user hand. But in case of RasMol, this is a neutral exploration of molecular structures, so there are no biases. Then rendering capabilities, so it has rich visualizations including space filling models, and it also provides us options for shadowing and reflective surfaces to enhance depth perception. Because if we are able to visualize the depth concept of the protein structure, then we will be able to know that which of the residues are exposed and which of the residues are buried, and which of the residues, because the residues which are kind of greatly exposed, so they are the sites which can interact with other proteins, whereas the residues which are lying inside or which are creating a pocket like structure, so they have different kind of functions. So depth can give us different information about the protein. So in this way, RasMol provide us that shadowing effect with which we can view the depth perception. Then performance improvements, so this was optimized shadowing algorithms for better speed and implemented on a single processor computers such as Unix, and later on Windows and on Macintosh also. So these are the operating systems. Then global usage, so it is widely used, especially in the educational setting, because many of the educational institutes use it because it is free of cost. It is cost-effective alternative to expensive molecular graphic softwares. So although there are molecular graphic softwares, but they are quite expensive, and there are some softwares which take a lot of time, because if you want to get a clear picture of the depth and all those things, so that will take some time, because in this case it is a quick method and it is also cost effective. Then CPK color scheme, so it is standardized coloring system which is used by the RasMol and Kind for elemental visualizations. So this is a CPK scheme, that which of the atoms will be colored by which of the color. So for example, in this case, the gray is used to represent carbon, white is for the hydrogen, red is for oxygen. So this is already defined scheme. So whenever you provide the protein structure in RasMol, you visualize it. So these are the colors which will define that what atoms or what are the elements which are present in the protein molecule. So this is already available color scheme. So for the uniform access, so anybody sitting anywhere will see only these colors. Then later on, protein data bank, which now stores all the protein three-dimensional structure information came into picture now, which provides us the information of the protein structures by the atomic coordinate files. So we can search PDB files by entering the PDB code, which is generally 4-letter. Currently it is 4-letter long. So in the search box, and we can retrieve the detailed structure information for the corresponding PDB entry. So in the previous sessions also we have seen that how if the structure is completely modeled either by X-ray crystallography, NMR or cryo-EM, so they can be uploaded to protein data bank and they can be stored so that anyone sitting anywhere can access them freely. So protein data bank is like a database which keep the information of all those structures. And now because it contains structure from different experiments, so it keep all the details that from where that structure is obtained. So it display the features of a PDB file. For example, if I search for a PDB entry here, then it will appear in this form. And now if we click on that entry, then it will show us different options. So by the end, we'll also look into PDB, that how we can search it. So now in display files, we can see this PDB file in different formats. We can see it in a FASTA sequence, we can see it in MMSIF format, or we can see it in the PDB format. So one by one, we'll see that what information do these formats provide us. But this is to show you that this PDB, it can give us information in different formats. So these are different formats, and then it also provides us some additional information such as the structural details, in which it will tell us about the atomic coordinates, that how different atoms are arranged in a three-dimensional space. So that is atomic coordinate, so it will provide us those details. Along with this, it will also tell us about the biological assembly. So generally the proteins are not always monomeric or a single chain proteins. In many cases they have multiple chains, and we have also seen that in some cases the PDB also have the structures along with their ligands or water molecules to stabilize those three-dimensional structures. So there is a biological assembly, so it will tell us about that biological assembly. Then primary citation, so it means that who has submitted it, so that information also we can obtain from PDB entry. And related literature, so if somebody has published something related to that protein, so all the publications based on that particular protein structure will also be provided by the PDB. Then it provides 3D visualization of the macromolecule. So as you can see in this case, so this is a 3D representation, and then further there are multiple tools in which you can open a PDB file and you can simply view that structure in different directions. You can move it, zoom it. So in this way, PDB also provides you the 3D visualization, and all these formats, they are downloadable. So as you can see in this, in display file, you can select any of the formats and you can also download those formats. So now one by one we'll look into all these file formats, that what information they keep and what are the syntax, what is the structure of these file formats. So beginning with the FASTA format. So in PDB we have a FASTA format, which is a text-based format, which will provide you a text-based information which represents the nucleotide or the amino acid sequences using single letter codes. So in this case, this is the further example of a nucleotide FASTA file. So in this case, firstly, the structure is that its first line will always start with a greater than symbol followed by the sequence name or comment. So there will be some sequence name or some comment to it, such as from which organism that sequence is adopted, or whether it is a nucleotide sequence or a protein sequence. So all those information will be given after this greater than sign. This will be the first line. Then comes the sequence. So sequence will be present in subsequent lines, but the condition is that in per row there will be characters equals to or less than 80. So if your sequence is greater than 80 characters, then it can take multiple lines, and ultimately it will end with the asterisk character, so such as star. So in many cases currently they do not use this asterisk as a to end the FASTA file. So in many cases you won't be able to see it, but previously in the format they used to put the asterisk symbol in the end of the FASTA file, so which represents that the sequence ends here. And this is the example of a multi-FASTA file in which there are multiple sequences which are present, and this is for the nucleotide sequence. Whereas if we talk about the protein sequence, so this is the FASTA file for the protein sequence. So what it tells you, firstly it starts with a greater than sign, then there is a ID. So this ID is a PDB ID, which is 9FAE_1. Then it also tells you the chain A, B and C, which means that this sequence is composed of 3 different chains. and then there are some comments such as it is a fiber and a human adenovirus 36, and then how many citations are available. So it depends that what information is provided by PDB, so that will be kept as a comment. And you can also create your own FASTA files. So one thing which you need to keep in mind that your file should start with a greater than sign, then you can write any comment, so that when you are aligning or you are doing some analysis, then you can figure out that which of the sequence you are talking about. So for that you can keep some comment, and then followed by the protein sequence, which is a one letter code. So this is the one letter code for all the amino acid residues present in the protein, arranged serially. So this is what information a FASTA file provides you. And now what are the advantages? So it is a simple and easy to process using text processing tools, and it is supported by Python, Ruby, Perl and other scripting languages. So one more thing, one more general knowledge information which I will provide you. So what you can do with the FASTA file of a protein. So this is a sequence based file, so it will not provide you any information about the structure. This will simply tell you what are the amino acids present in your protein. So now how it can help you. For example, you are working in a laboratory where you identify the new sequence, and you want to figure out that which organism or which microbe you have identified, because by looking at that sequence you cannot tell directly that which organism is it. So by using this FASTA file you can perform different alignments, such as you can go for BLAST. So BLAST is a tool with which you can align this sequence to available protein sequences which are already available. So by performing protein BLAST, it will check that to which other available protein sequence it aligns best to, it matches to. So with which it can tell you, just give me a minute. Okay, so I hope now it is shared. Yeah, so means with the help of this particular FASTA format, you will be able to know that which organism this particular protein sequence belongs to. If it already have the same sequence already available in the BLAST database, or if it is unknown sequence or it is a novel protein sequence, then also you can figure it out. So that is one of the advantage that why we go for the FASTA format. Then the second format is the CIF format, which is known as crystallographic information file. So this is a standard text file format which represents the crystallographic informations established by International Union of Crystallography, and it tells you the full specifications of this is available at this IUCR website, and it is compatible with molecular viewing programs like PyMOL. So this is the representation that what information the CIF file provides you. So it tells you about the symmetry, that what is the symmetry of the unit cell. So it is triclinic in this case. What is the space group? So it tells you about the space group name. Then it tells you three different parameters, which is ABC, which is the length of the unit cell on three directions, and then it also tells you the angle between these three sides, such as alpha, beta, gamma. And along with this, it also tells you about the cell volume. So this is the information of the unit cell which is provided by this CIF file. So it is generally providing you all the information of the X-ray crystallographic experiments. Then comes macromolecular crystallographic information file, which is MMCIF. So this is the latest version of CIF file. So when you will go to PDB, then you will see that currently MMCIF version is available there. So this is the advanced version. So in addition to the CIF format, in which you only get the information about the unit cell symmetry and unit cell structure, in MMCIF you will also be able to know detailed information, such as you will also be able to see the journal name, the citations, journal volume, page first, who have submitted it. So all those additional informations are also provided by the macromolecular CIF file. So this is alternative to the PDB format, and now it is the default format in PDB. It is flexible, extensible, tag value format which is used for the macromolecular structural data representations. Then the next format is the MTZ format. So this stores the electron density maps from experimental data. Loading an MTZ file is slow since the map needs to be calculated. So an option exists to recalculate with different grid spacing for better image quality. So these are known as grids. If you will keep the grid spacing to be very low, then it will take a lot of time, and if this grid spacing is high enough, then it will take less time. But it depends that how do you select it. Generally, calculation of electron density maps is time consuming. So this is how the electron density map appears, and this is the MTZ format because it requires selecting the structure factor and phase, which is phi, column data to compute the map. And the default grid size is 0.75 angstrom, which is suitable for model building, and smaller grid size gives higher resolution but slow performance. So as I was telling that these blue lines, they represents the grid sizes. So the space between these blue lines tells you about the grid size. So if this size is very small, then although it can increase the resolution, but it will take a lot of time. So we need to make a balance between both of them and decide the reasonable grid size, which is 0.75 angstroms. So if models are loaded, users can associate them with the map, treating them as a part of a crystal structure. Yeah, so this is how you can calculate the electron density maps by taking your MTZ file, and you can open it, and this is how it will appear. Then the very important thing in PDB is the validation report. So whenever a person submits his three-dimensional structure or a protein structure to PDB, so there need to be the generation of validation report, which will verify that whether the structure which is crystallized or the structure which is submitted, it is valid or not. So for example, if we compare these two structures, so in validation report there are different measures with which it identifies that whether your structure is valid or not. So in this case, structure A and B, A is relatively good whereas B is poor. So now how it is poor, these are different metrics with which we judge. So if your values lie towards the red color, which means towards the left, which means it is a worse, and if it lies towards the right, then it means it is better. So first one is R free, then clash score, so which checks that whether there are some steric clashes or not. Then Ramachandran outliers, so whether the amino acids they are lying in the, for example in Ramachandran plot, it checks for the phi psi angles, so whether those dihedral angles, whether they are favorable for a particular structure or not. So in Ramachandran, if it is outlier, then that is also a problem. So you need to be, outlier value should be as low as possible. Then comes side chain outliers, then RSRZ outliers, RNA backbone. So if its value is towards the blue side, then it is a better structure, and then there are chances that it will be accepted and it will be available through PDB. Whereas if this is how its metrics appear, if most of the values they are lying towards the red region, then it means the structure which you are submitting, it is worse. So in that case, PDB will tell you that you need to refine your structure. So then you have to go through those refinements, and you have to identify that what are the reasons which are leading for these problems, these outliers. So in this case, if we look for the sequence based representation in the validation report, then in this case these red dots, it tells you that which of the amino acids they are lying in the invalid region. Whereas in this case, if you look at the B structure, so there are multiple such red dots, which means that multiple of these amino acid residues they are lying in the unfavorable region, due to which we observe the values towards the red side, or we observe the greater outliers in the case of structure B. So this is how, with the help of validation report, we can compare that which PDB structure we can take and we can accept and we can study. So if we look here, so these are the residue sequence plots and outliers, which flags the residues with unusual geometry. Now how the geometry can be unusual? So there are different parameters such as bond length, bond angles, Ramachandran, RNA smoothness, torsion angles. So if these angles are not favorable, so that tells you that the structure is unusual. So you need to refine it further. And these colors, so green tells you that there are no geometric outliers, yellow tells you that there is one type of outliers, orange tells you that there are two types of outliers, for example in these cases, and red tells you there are more than three plus types of outliers in this case, because these are the different outlier types 1 2 3 4 5 6. So if there are multiple such outliers, so which tells you that that particular residue is invalid, or it is unfavorable, highly unfavorable at that particular position. Gray tells you that there are no atomic coordinates available. Cyan tells you that three defined atomic coordinates NMR ensemble. That means in certain scenarios what can happen is that the occupancy of that particular residue can vary. So in that scenario we have different atomic coordinates, we have an ensemble of that coordinates, so that is represented by the cyan color. So for X-ray structures, a red dot above a residue indicates poor electron density fit. So these red dots, they tells you that there is a poor density fit, but these green colors, they tells you that there is no geometric outliers. So you need to make sure it is also possible that it favors one of the metric, but it is not favoring the other metric. So we need to check overall that what is the overall metric score, so by which we can validate the protein structure which is submitted to the PDB. Now the main thing is that how we can read a PDB file. So in this case, PDB file, we are talking about the PDB file format. So till now we have seen the FASTA format, we have seen the CIF format, we have seen MMCIF format, and we have seen the MTZ format, which is the electron density map, and then we have checked for the validation. So now we'll see the main format, which is the PDB format, which generally tells you about the three-dimensional information of the protein. So in PDB file format, each line is 80 columns wide, ending with the semicolon. So each line will end with a semicolon, and it can be up to 80 columns wide. First 6 columns, they tells you about the contains a record name which must match the specific PDB record type. So for example, this tells you about the header, that is what is the type of molecule. It also tells you about the date of release, its PDB ID, then what is the molecule name. For example, if it is a transferase, is a class of enzyme. So in this case, it also tells you that what is the name of that particular molecule, which enzyme it is. So it is telling you that it is deoxycytidine kinase. Then it also tells you about the chain and how many number of chains are present along with their name. Then it also tells you about the organism from which that particular protein was taken. So in this case it is Homo sapiens. Then it tells you about the common name of that organism. So in this case it is human. It also tells you about the taxon ID, which is 9606 for the human. So all these information are present in the PDB file. So firstly it tells you about the detailed description of that particular protein, and later on it tells, after this description, it tells you about the journal in which it is published and what are the different literatures, different citations which are available for that particular protein entry. Then it tells you about different remarks such as its resolution. So whenever we select a PDB file for our study, so we also check that what is its resolution, because if it is of high resolution, then we tend to favor those structures which have high resolution. So it also tells you about the resolution, and later on it tells you about the coordinate information of particular protein. So now let's look one by one. So these are the key information in the PDB file. So we have seen that it provides type of molecule, date of release, PDB ID, molecule name, number of chains and chain names, then scientific name of the organism, its common name, scientific journal details, includes authors, title, reference, DOI and PMID under journal. So this information is also important because sometimes if you have a PDB structure, and for example, if I am working in a lab and I have performed X-ray crystallography on certain protein and I have submitted it to PDB, and now in validation report my protein failed, it is not supporting all those metrics, and that structure is not favorable. So in that case, what can happen is that in PDB that same protein can also be predicted by some other scientist. So what I can do is that I can search for that protein which is already predicted, and then I can check by looking at this PMID. I will open up their paper and I can see that what analysis they have done. or what they have done, how they have operated on it. So with which I can gain some understanding. So for which this PMID is very important. This citation or journal information is important, and so that I can understand that how they have performed crystallization. Or for example, if I want to know that what are the different conditions, or from which organism they have isolated it and how they have performed the extraction, so all those things we can study from these journal details. Then structure resolution, so this is also important because of the resolution is, if it is high resolution, then the structure is much more better. We'll consider those structures which have high resolution for performing our analysis. Then comes unit cell parameters in PDB files. So these unit cell parameters, it tells you about the crystallographic information. Okay, so sorry, there's a lot of network issue today. Yeah, so now we'll see the unit cell parameters in PDB files. So it tells you about the unit cell information. For example, if you have performed X-ray crystallography, so in that way it will tell you about the unit cell parameters. So automatic validation check, the coordinate file must contain crystal1 record to define the unit cell and space group information. So if it is a crystallographic structure, then this information will be provided, such as the record of that atom, its serial number, residue name, residue serial number, XYZ coordinates, occupancy, thermal factor, and the element symbol. So element means, for example, in arginine we can have different elements such as nitrogen, carbon, oxygen, alpha carbon, beta carbon. So all those are different elements which are present within a single residue. So this is the element symbol, because each atom or each element can have different XYZ coordinates. So because a residue is a combination or collection of different elements, so that's why we have the information at the atomic levels. So these are the atomic coordinates, and along with this, each PDB file will also have a crystal1 record in which it will tell you about the length of ABC, which is the unit cell length along these three directions, along with the alpha, beta and gamma angles, along with the space group name if it is obtained from the X-ray crystallography. But if it is a non-crystallographic structure, so if it is not determined by crystallography, then it is assigned the so for non-crystallographic structures, if the structure was not determined by crystallography, for example, if NMR or cryo-EM has been used for predicting that structure, so it is defined, it is provided, or it is assigned with the default unit cell in which ABC will be 1, alpha, beta, gamma will be 90 degrees, and space group will be P1, which represents a simple cubic unit cell. So in cases where you find simple cubic unit cell, so you can understand that this particular structure has not been resolved by X-ray crystallography, either it has used NMR or it has used cryo-EM. So this ensures the compatibility with the validation tools even if we do not have the crystallographic data, so that we can maintain the format of a PDB file even if we do not have the crystallographic data. So for that reason, for the compatibility, we still keep the crystal1 record even if it has not been solved by the X-ray crystallography. Then comes TER, which represents the termination. That means wherever your protein terminates, your protein sequence terminates, so it will be given by the TER. So in this case, TER shows that here your chains, all the protein chains ends. So after this, there will be another record which is heteroatom. Heteroatom means what are the different atoms such as water molecules or some ligand, because in many cases in PDB we provide a complex proteins in which the proteins are already in complex with certain ligands or water molecules, so they are represented in the heteroatom after the TER record. So it marks the end of an atom record for a polymer chain. For proteins, the residues listed in the TER record is the carboxy terminal residue of that chain. So you will see that before it, the residue which will be mentioned, that will be a carboxy terminal residue. Then came heteroatom. So which the purpose to define the heteroatoms is to give you the information that what are the different known protein atoms which are present in your protein structure, so such as water molecules. There could be some metal ions or ligands. So these ions, water molecules, they are generally used to stabilize the structure, because when we perform crystallography, so in certain scenarios we need to use these atoms so that your structure is in a near native state. Because in our bodies the protein is covered, or it is present in the aqueous environment, so to replicate that same conditions we add the water molecules. So that is the reason we have the heteroatoms in our PDB file. Then is the another record after the heteroatom is the connect. So connect tells you that what are the different bonded interactions, different angles, different dihedral angles between atoms which are present. And the usage is, it defines the intramolecular bonds. It specifies the interactions between the protein atoms and heteroatoms. For example, the metal coordinates. So as you can see here, so these are the values. These are the different values which tells you that between which of these positions, between which of these elements there is some kind of interaction, interaction such as the metal coordinations or all those things, or the intramolecular bonds. Or you can also see that if there is some dihedral angles present between two different elements, then all those informations will be given by the connect. Now we can perform different calculations on our PDB file, such as we can perform distance calculation. Now why distance calculation? Because by looking at the distance, we will be able to figure out that how far the two elements or two atoms are how far to each other. Because if the distance between the two atoms, if it is less than 3.5 angstrom, then there is a possibility that they can make hydrogen bond, they can interact. So to study those things, we can perform the distance based calculations. So how we can identify the distance between the two atoms? So for atom 1, which is present in the atomic coordinate x1, y1, z1, so this tells you that in the 3D coordinate space where the atom 1 is located. Similarly, atom 2 will also have a single point value towards x, y and z axis. So there is only a one point towards x, y and z axis which tells you this information. Now to find the distance, the formula is square root x1 minus x2 square plus y1 minus y2 square plus z1 minus z2 square. So this will tell you the distance between atom 1 and atom 2. So one thing you can understand from here is that to find the distance between two atoms, it is very important that you must know their xyz coordinates. Then comes angle calculations. So if you want to find out angle between different atoms, or if you want to calculate the dihedral angles, so for those things also you need to know the coordinates. For example, if we have P1, P2 and P3, which are three different coordinates, and we want to find the angle between them, between P1, P2 and P3. So this is a plane. If you want to find the angle between this plane, so what we can do, we have the xyz coordinate for P1, we have xyz coordinate for our P2 point, and we also have xyz coordinate for our point P3. So now we'll calculate A and B vectors. So A vector will be P2 minus P1, B vector will be P3 minus P2, which is calculated by this formula. And now we can calculate the angle theta, which will be cos inverse of dot product of AB and then their product of their individual magnitudes. So this is how we can calculate this angle by having the towards this plane P1, P2, P3. So this is how we can calculate the angles. Now we can also identify the dihedral angles, because from the PDB file we perform the because in PDB, because Ramachandran and all those validations need to be performed. So for that, dihedral angle calculation is also important, because after getting the xyz coordinates, we also make sure to validate that we calculate the dihedral angles, that either they are favorable or not. So in this case, cos phi can be calculated as Na dot Nb divided by product of their magnitudes Na and Nb. So this is the dot product. So dihedral angle is the angle from the plane containing A and B to the plane containing B and C. So this is one plane and this is another plane. So it is the angle between these two planes, and it is the angle from the normal vector A cross B, which is Na to normal vector B by C, which is Nb, and counterclockwise around B, where A cross product B and B cross product C are the cross products. So in this way, these are the dihedral angle calculations, which is the angle formed between the two planes by finding out the normal and then identifying that value. So this one is the phi, and these are the two planes. So these two planes, so this is the normal drawn to these two planes. So this angle is phi angle, which we calculate in the dihedral angle calculations, and this is the formula. So this is to show you that these are the different calculations that can be performed on the PDB file. And now we can also predict the secondary structures from PDB file. So criteria for the alpha helix. So a hydrogen bond is formed between the residue I and I plus 4. So this is the condition for the alpha helix to form. So in alpha helix, we always observe the hydrogen bond between the first residue and fifth residue. So we need to identify the residue pairs I and I plus 4, where the oxygen and nitrogen atoms, they have a distance less than 3.5 angstrom. So if this distance is less than 3.5 angstrom, then there is a possibility that there can be formation of hydrogen bond between I and I plus 4 residue, or I and I plus 4 residue. And then we continue checking for these residues. Then we'll proceed it. So firstly we'll check for I and I plus 4. So if we find out that distance is less than 3.5 angstrom, then we will continue our search by adding 1 to I. So that means if we have started with residue 1 and then checked 5th, then we'll continue it for residue 2 and then 2 plus 5 and 7th residue. So similarly we'll repeat it until and unless the distance between those two residues increases 3.5 angstrom, which will tells us that now the helix has stopped or the helix breaks here. Then how we can identify the beta sheet? So in beta sheet, there is a hydrogen bond which forms between the residue I in the strand 1 and residue J in the strand 2. For beta sheet, we can observe both the parallel and anti-parallel beta sheets. So the condition for parallel beta sheet is that the residues I plus 2 in the strand 1 binds to J plus 2 in the strand 2. So for example, in this case, these are the parallel beta strands. So the residue which is I plus 2, it binds to the J plus 2 in the strand 2. So in this way, if such condition is met, if the distance between these two residues is less than 3.5 angstrom, then it means this can form a parallel beta sheet. So we'll continue the search till the point the distance exceeds 3.5. So that will tell us that where the parallel beta sheet ends. And then there is another condition. So for example, if we observe that residue I plus 2 in strand 1, it is binding to the J minus 2 in the strand 2. So this tells us that this is a anti-parallel beta sheet in which the directionality of both the running beta sheets, they are anti-parallel to each other. So based on these calculations, we can also predict that whether it can take a beta sheet structure or whether it can take a alpha helical structure. Then we'll talk about COOT, which is known as Crystallographic Object Oriented Toolkit. So this is a tool which is used for the macromolecular model building, completion and validation, particularly for protein modeling using experimental structure determination data. So for example, if we have a PDB file, then we can simply open it in COOT and we can analyze it. So not just PDB but MTZ file, all different, there are different formats which are supported by COOT. So we can open our macromolecular structure, we can build it, we can perform validation of that structure in COOT. So it provides us electron density map visualization and it enables the model manipulations. So we can manipulate our protein structure as well in COOT, such as we can perform idealization, we can perform real space refinements, we can perform manual rotations and translations. So in that structure, we can rotate it, we can translate it, we can perform different things. We can perform rigid body fitting, we can perform ligand search, we can also perform the solvation, mutations, rotamers, Ramachandran plots, we can perform non-crystallographic symmetry adjustments. So it is designed for ease of use. Just give me a moment. Yeah, so it is designed for its ease of use, featuring the intuitive user interfaces with menus, toolbars, and mouse controls. It is a free software originally from University of York, and now it is maintained at the MRC Laboratory of Molecular Biology. So this is how it represents the electron density maps in COOT, and this is the diorama in which you can see the Ramachandran plot. So this is for the validation. So if you have a structure which is available, then you can validate it. You can find it out that either it is lying in the Ramachandran favorable region or not. So in this case, these pink bars, they are lying in the Ramachandran favorable region or allowed region. So which means that the structure is valid. Along with this, COOT will tell you about the different file compatibility. So COOT can read the 3D coordinate models in different formats such as PDB, MMSIF, Shelx files. So these are different files which it supports. 3D visualizations. So models can be rotated, viewed from many angles. So if you go to COOT, then you can open your PDB file and you can rotate it. You can simply use the mouse to rotate it and view it from different angles. So that flexibility COOT provides you. You can use a stick model representation where vectors represents the chemical bonds and bond halves are colored by element types. So as in this case, you can see this is the stick representation in which all the residues present in the protein, they are represented by sticks, and these different colors, it tells you that which are the different chemical bonds or which are the different elements such as oxygen. If oxygen is present, it is represented by red. If nitrogen is present, represented by these blue colored sticks. If it is a carbon, or if the carbon element is present, then it is represented by these yellow lines, and the water molecules are represented by these red dots. So means with this you can see that by looking at it you can simply figure out that which of the element it is showing you. So they are differently colored. Then electron density display. So it shows you electron density maps from X-ray crystallography and EM reconstructions. So density is contoured using a 3D mesh for better visualization. Model building and validation. So it offers you tools for model refinement, adjusting structures to fit the electron density better. It ensures that the atomic models align with experimentally derived electron density and follow chemical rules. So these are different model building and validation also, which is provided to you by the COOT. Now we'll look into different applications of this tool, which is COOT. So first of all, the major application is that it is user friendly. So it has a smooth interface, it is easy to use. It is interactive, 3D visualization for model manipulations, intuitive navigations for structural biologists. So first of all, because you can simply open your PDB file by clicking on the file open PDB, and now it's quite flexible. It is GUI interface, graphical user interface. You do not have to enter different commands, or you do not have to remember anything. You can simply view it, you can move it, you can see it from different angles, you can manipulate it. So with those reasons, it is user friendly. Then comes powerful features. So it supports real-time model building and refinements. So in the real time only you can manipulate it, you can refine it. So such as these are some of the options. For example, you can use torsion restraints, you can use planar peptide restraints, you can use trans peptide restraints, you can use Ramachandran restraints. So if you have a model which is having certain psi angles, so by putting those restraints, you can try to validate it, or you try to refine it kind of. So if you click on these options and if you click OK, then it on the real time, at the same time, it will perform these refinements and it will give you the structure after the refinements. So that is known as real-time model building and refinement. It allows fitting and editing atomic models into electron density maps. So you can also look the electron density maps for that particular protein model. Then comes automated refinement tools for structure correction. It also provides you ligand fitting tools to analyze the small molecular interactions. So these are some of its powerful features. Then comes functional analysis. So it assists in understanding protein ligand interactions. It facilitates the mutational studies by modifying residues. It helps in validating and correcting the macromolecular structures. So these are some of the functional analysis which we can perform using COOT. Then if we talk about its compatibility, then it is compatible with CC4, Phoenix and other crystallographic softwares. So these are different softwares, and it also supports multiple file formats such as PDB, MMSIF, MTC. So all these file formats we can open in COOT and we can perform analysis, validations and all those things. And it can also be integrated with Python scripting for automation. So for example, if you have multiple such PDB files and if you want to analyze it, so in that case you can also use the Python scripting. So it can be integrated with scripting also to automate it. Then real-time feedback and validation. So it provides you real-time electron density map adjustments. It highlights the geometry errors using validation tools. It also supports the interactive Ramachandran and rotamer analysis. So in the real time only you can perform these analyses as well. And it is open source and free, which is one of the major advantage. So it is available for Linux, Windows and Mac OS. And it is open source, so due to which the community can simply use it. So and it can also be improved because the community which is using it, they can provide feedback. So in this way it can be improved. Now before looking into the applications of COOT further, we'll firstly talk about the case of beta-lactams, which are generally used as antibiotics. So beta-lactams, how they have the antibiotic properties. So when we say antibiotics, that means they are inhibiting the growth or they are killing the microbes. So the mechanism of these beta-lactams, such as penicillin is the example of a beta-lactam because it has a beta-lactam ring. So it inhibits the peptidoglycan synthesis, weakening the bacterial cell walls. So as we all know that in the gram-negative bacteria, we have in their cell wall they have peptidoglycans. So in which there is a NAG-NAM polymers, and which these NAM they have the pentapeptide, which is the peptide composed of 5 residues. So these are 5 residues. From both the sides, these peptidoglycan. So from both the sides, this N-acetylmuramic acid, it contains the pentapeptides, among which the bond is formed between the L-lysine and D-alanine. So by this bond, one D-alanine is removed. Similarly from here also, the bond will be formed between D-alanine and L-lysine, and D-alanine will be removed with the help of this enzyme, which is known as PBP, which is penicillin binding protein. So this particular enzyme is responsible for cross-linking between these 2 layers of the NAG and NAM, these 2 layers, and performing the cross-linking with the help of the pentapeptides. So this is the PBP, which is penicillin binding protein. Why it has the name penicillin binding protein? Because it can also bind to penicillin. So in the normal scenario, when penicillin is not present, then in the bacterial cell wall, the cross-linking is happening, which is maintaining the rigid structure of the bacterial cell wall. But as soon as you provide the penicillin, so in such scenarios, what will happen that now this PBP, it will bind to penicillin, and now it will not be able to bind to DLR and perform the cross-linking. So if there is inhibition of cross-linking, then the cell wall will rupture. So bacteria will eventually die because his inner content will come in contact with the external environment or external substances, due to which bacteria can die. So this is how the antibiotics behave, and penicillin is the example of beta-lactams. And then another thing is that these DLR, this is the structure of DLR, and this is the structure of penicillin. So they both look like the mimic of each other. So due to which PBP gets confused and it binds to penicillin because it both have the similar structures. So then comes the beta-lactamase. So whenever the mase is there, it tells you about that it is an enzyme. And whenever it is written as beta-lactamase, so that means the enzyme which degrades beta-lactam. So in this case, beta-lactamase, it breaks the beta-lactam ring, which inactivates the penicillin. And structural analysis of this beta-lactamase before and after the reaction is captured in PDB and MTC file. So if this structural analysis is captured, then we can use these PDB and MTC files and we can open them with COOT, and we can understand that how the structure is changing, what are the different things which are happening at the level of structures. So this is one of the advantage of COOT, that we can see in the real time that how the structural changes brings about the functionality changes and what are the things which are taking place inside the protein. So in this case, as we provide the beta-lactamase, So now what is happening is that in active penicillin we have a beta-lactam ring. So in the presence of beta-lactamase, there is this breakage or hydrolysis between the bond which is present between N and C double bond O. So this breaks and this inactivates the penicillin. So due to breakage of this beta-lactam, now penicillin is no longer able to bind to PBP. So in such scenarios, now the bacterial cell wall can perform the easily cross-linking. So due to which, this is also one of the evolution which has taken place in different microbes. So now they have ability to produce beta-lactamase. So they have generated resistance towards different antibiotics. So the key finding is that the conformational changes in beta-lactamase observed and relevant for the drug design to combat resistance. So by looking at these conformational changes and all those things with the help of COOT and different protein visualization tools, now we can understand that how the conformation is changing. So we can design new drug molecules and we can see that whether those new drug molecules can bring about change in the conformation of the protein or not. So these are some of the advantages. Now we will see visual representation, that how this is behaving. So this transpeptidase, also known as penicillin binding protein, which is represented by this green structure. So this is showing you that how it is binding the two layers of NAG-NAM by this pentapeptide bond. So this is the normal scenario in which the transpeptidase is behaving normally without the presence of beta-lactam. But as soon as if you have a beta-lactam, so this is the beta-lactam, which is the penicillin. So now what can happen is that this PBP, penicillin binding protein, instead of binding to DLR of this particular pentapeptide, it will bind with the beta-lactam. So now there will be no reaction between these, or there will be no interaction between my PBP and this pentapeptide. So cross-linking will not take place in the presence of beta-lactams, which can disturb my cell wall. So in this case you can see there is a rigid sturdy cell wall, but in this case cell wall can rupture. Now what can happen in the presence of beta-lactamase? So this is my beta-lactam and this green structure is my beta-lactamase. So it is performing hydrolysis, it is breaking the beta-lactam structure, this is breaking the beta-lactam ring. So now due to breakage of this ring, it cannot interact with PBP. So in such scenarios, my PBP can behave normally, it can perform carboxypeptidation in which it can perform the cross-linking. So this is just the visual representation of what we have seen so far. And now there are different types of beta-lactamase, class A, class B, class C, class D. And these class A, C, D, they have serine in their active sites with which they behave. And then in case of class B, they have some zinc molecules which are present and in the active site. So if we talk about beta-lactamase class A, now we are seeing that how the chemical reactions are taking place, how it is breaking my penicillin ring. So in this case, just give me a minute. So in this case what is happening is that if we take the example of class A of beta-lactamase, then in this case it has serine in its active site. So one by one we'll see. So first initial interaction is happening, which is leading to acylation. So the serine residue which is present at the 70th position in the active site, it acts as a nucleophile. So it will attack this beta-lactam ring, which is a 4-membered cyclic amide of the antibiotic. Now this will break the amide bond which is present between C double bond O and this NH. So this amide bond will break, which will lead to the formation of acyl enzyme intermediate. So this is the intermediate which will be formed. Now this R3 group from the beta-lactam, so these are different groups, R1, R2 and R3. Now this R3 group is eliminated and the proton is released. So this R3 will be eliminated. Now the intermediate stabilization will take place in which lysine 73 and glutamate 166, they helps to stabilize this transition state by performing the charged interaction. So now they will perform charged interaction. So as we know that glutamate is negatively charged and lysine is positively charged, so they will tend to stabilize this intermediate structure. Now this enzyme will remain temporarily covalently bonded to the substrate, and now in the end there will be deacylation in which The water molecules, which are assisted by glutamate 166, they hydrolyze the acyl enzyme complex, and this will lead to regeneration of the active site and complete breakdown of this antibiotic. This inactivated antibiotic is released and beta lactamase is free to act on the other molecules. So eventually this particular beta lactamase, it will be removed from this particular beta lactam on which it was attacking. So in this case, for easier representation, we are only showing you the 70th residue, 166 residue of beta lactamase, but in generally there will be a huge protein which is interacting with this. But in this case, only the residues which are interacting, only they are highlighted to for the ease of viewing. So that is the one thing. So in the end, after breaking down this beta lactam structure, the beta lactamase, it will separate from this particular substrate. So this is leading to inactivation of my beta lactam or penicillin. Now we will talk about the molecular visualization platforms such as PyMOL. So in WinCOOT also we have seen that we can perform different things. So PyMOL is also one of the easier tool in which we can visualize the protein molecules. So it is a powerful open source molecular visualization tools which is widely used in structural biology, drug discovery and bioinformatics. So it allows the researchers to visualize, analyze and manipulate three-dimensional structures of proteins, nucleic acids and small molecules. And it supports high quality rendering, molecular docking analysis and electrostatic surface mapping, making it a valuable tool for studying biomolecular interactions. And its scripting capabilities also enhance its automation and reproducibility in research. So now we will see that how its main page looks like. So in PyMOL, as soon as you download this app, so you will be able to open it. So when you will open it, you will see different sections of this particular app. So first of all, it tells you the pull down menus in which you can click on file, edit, build, movie, display, settings, scene, mouse, wizard, help. So these are different menus which are provided you by the PyMOL. So one more thing which I want to add, that because each time the app gets updated, so you might not be now currently see this kind of view. It might get changed, and for different systems or different operating systems such as in Mac, in Windows, in Linux, you can see different ways it might appear. So this is the pull down menu in which you will be able to click on file and you will be able to open your PDB file for visualization. So these are known as pull down menus, and this will read that file in the text format. This is the command pane. So for example, I uses the Mac OS. So in that case, PyMOL appears in the bottom. So in some scenarios, PyMOL appears in the top, in which you can enter the command. So this is known as the command line or command box, in which you can enter the command. If you do not want to use these visual options, then you can use it by the commands also. You can perform analysis. This one is the sequence pane, in which you can view the sequence of that PDB file, that what is the sequence of amino acids which are present, and if there are some ligands, water molecules, and then they will also be operated. So the advantage of this sequence pane is that you can select the desired sequence, you can color it accordingly, and you can uniquely study it, you can separately study it. So you can perform that analysis easily, selection, because it's quite difficult to select the chains or different residues from here. So the sequence pane make it easier, the selection process. And if the sequence is not visible when you open your file, then there is a small S letter which is written here. So if you click on it, it will show you the sequence pane, and you can also modify it. So for example, if you go to display, then from display you can decide that what the sequence pane should show you. It can show you amino acid residues, it can also show you the chain names. So in that way, it depends that what do you want to see with the help of sequence pane. Then comes main window, which will show you the three-dimensional structure of the protein. So for example, if I open a PDB file, then its three-dimensional structure will be shown in the main window, and in the object pane, it will tell you the ID, PDB ID of that particular protein structure. So in this case, it is 1HEW, and all means if you perform different selections or different things on 1HEW, if you want to calculate electrostatic interactions, or if you perform different analysis, so in that case multiple such smaller sessions will appear. So in this all signifies that how many such analysis you have performed. So 1HEW is the PDB ID which you have opened here. and then there are different options. So one by one we'll see them in the later slides at what each option means. And here you have a mouse control from where which tells you that what are the different controls which are present to you with the help of mouse, different selections or different right button, left button, click. So it tells you different controls with the help of mouse. And then these are known as movie controls, which are present in the end. So with which, if you want to make a movie, then also you can create a movie in which you can select different frames, you can move it here and there and then capture them in different frames and make it smooth so that it appears like a movie, that it is naturally moving. So that also we'll see in the subsequent slides, that how you can do these things. So in this way, it allows the researchers to visualize, analyze and manipulate the three-dimensional structures of proteins, nucleic acids and small molecules, and these different things that it is supporting high quality rendering, docking analysis. So molecular docking means that if you have an enzyme, then you can dock it with some suitable substrate or suitable ligand. So it is helpful in case of drug designing, so that you can see that where your ligand or where your substrate can bind to the enzyme or a protein, so that you can make some changes so that it binds better. And then electrostatic surface mapping, so all those things can be done in PyMOL. Now we will move into the detail of PyMOL. So first of all, let's talk about opening a new structure. So how to open a structure in PyMOL? So easy way is that you simply go to file, you click on open and you choose a file. But before this you must have the PDB file. So for that you can go to PDB and you can enter the PDB ID, you can download that file and you can simply open it. And then as soon as you click on open and you select the file name, it will open up. But in some scenarios, if you do not have the PDB file but you know the PDB ID, so now PyMOL provides you the advantage that you can simply write in the command box fetch followed by the PDB ID. For example, in this case my PDB ID is 1HEW. So I have entered this command here and clicked enter. So as soon as you hit enter, that particular structure will open up, and this is the command pane in which it will tell you all the commands which you have entered. So for example, in this case PyMOL fetch, this command I have already entered, so it is coming here, but you have to write it here and then click enter. So then it will be in this console, it will be shown here. So this is how you can open any new structure in your PyMOL. Now you can also set different colors for your structure, such as in this case it is shown in green color. So I can also change, I can select in which color I want to see it. So in this case, these ball and stick, this is a ligand and this one is 1HEW protein. So in this case, this is a complex protein which is studied or which is crystallized in the presence of a ligand to see its interactions. So in this case, I can also decide that my ligand should appear in different format and my protein should appear in different orientation, or you can say my protein should appear in different representation. So one by one we'll see that how we decide it. So in this case we can select the RGB color. So if you simply want to color it, then you can either select from these options, or otherwise you can also enter the command. So in this case the command is set color hey followed by square bracket, and these are three values which are entered. So these belong to RGB values. So R for red, middle one is for green and last one is for blue. So depending on what is maximum, that will decide the color which your protein will get. And then to run this command you have to type set cartoon underscore color hey. So now this will apply the color to your structure which you have loaded. So now I'll show you that how it works. So for example, we have this protein structure which we have loaded in PyMOL. So now I can type this command. I can select, depending on my choice, I can write any color inside the square bracket. So these are three values separated by comma. This will tell you what color it will take, and then I can set this color. Now as soon as I will enter it, this is how my color will change. So in this case, because my central value was highest and other values were 0, 0, so which means it will be more green, RGB. So G is in the middle. So highest the value in that particular block, it will determine the color. So this is how the color will change, and we can also change it to other color depending on what is the RGB code. So for example, here is a table in which I have provided the information. So those who are interested, they can open the PyMOL and they can open the PDB file. So you can change the value of these which are written in these square brackets. So by changing these values, your color will also change. And this is the command based way to change the color, although there are some other ways also from these options which are given. So C means color, so by this also you can change the color. And along with this, you can also show the electrostatics, that whether the residues present in your protein, either they are positively charged, negatively charged, what is the concentration, which residues are in greater abundance. So that electrostatic analysis can also be done. So for that you need to go to wizard. So in this case, this is the wizard menu. If you click on it, then it will open a bar in which it gives you different options. So now we have to go to demo, and as you click on demo, it will show you different demonstrations such as representations, cartoon ribbons, volume rendering, roving details. So if you have time, then you can click on these options and you can see that how it changes the format of a molecule. So now if we click on electrostatics, then this is how it will show the electrostatics for my protein molecule. So I started with 1 HEW. So for that, this is the electrostatic. So these red colors, it tells you the intensity of negatively charged residues, and blue represents the positively charged residues. So in this case, as you can see, the intensity of red is quite high, blue is less, so which means that your protein, it contains the most of the negatively charged residues. So why this electrostatic study is important? Because it will give you some idea that because you are performing analysis, so you will be able to know that to which other proteins my given protein can interact with. If it is negatively charged, then there is a possibility that it will interact with those substrates which contain positively charged residues, or those proteins which contain positively charged residues. So this is one of the advantage of looking at the electrostatic values. Okay, sorry. Yeah, so now let's move to basic visualization and interaction. So this might contain certain things which we have already seen, but this is for the general knowledge of basic understanding that what are different things which are shown to you in the PyMOL. So first is initial view and structure representation. So the default molecular representation, it appears as a green, blue, red line model. So as we have seen in this case. So this is how it appears, which do not tells us, which confuses us. So with a black background, so this is a row representation which is not useful for identifying important structural features. And as I have also talked that in that case 1-HEW, it also has an inhibitor in it, which is N-acetyl glucose, which is present, but it is hard to spot in my first structure. So how we can spot it? We need to select certain molecules, we need to decide how to represent it. So with that, it becomes much more easier to study it. So navigating the molecular structure, if we open it in PyMOL, then we can rotate it. And how we can rotate? We can use the left mouse button while moving the mouse. We can zoom it. We can also perform the slab view, which uses the mouse wheel to scroll and adjust the view inside the molecule. Now let's understand this object panel. So this object panel command, it is in the right hand side of the PyMOL interface, and it provides the mouse based commands for easy interaction. So these are different keys: left, middle, right, wheel. So this tells you that what are the different things which can be done with these keys, and this tells you that if along with shift, if you click on left button, then it will add box, minus box. So this is the mouse control. Now comes the main buttons in the object panel. So because we have seen in the object panel, let's go here. So this is the object panel which tells you, so yeah, so here this is the object panel. These are some of the buttons and these are the mouse controllers. So mouse controllers we have seen. Now we'll see that what are these buttons, what are the advantage of these buttons, and what are the different analysis we can perform. So A stands for action. So with A, you can perform various actions, you can perform analysis and calculations. So in later slide, we'll see what are the different options you can get from this action button. Then comes second, which is S, that means show. So it applies specific graphical representations to molecular structures. So you can define what to show. Then comes hide, H. So H tells you that what you want to hide. For example, if you have both the protein chain and the ligand and you want to hide the ligand, as in our case we have NAG, which is N-acetyl glucose, so we can hide it. And if you want to simply, we can simply study our protein. So that hide provides you that advantage that you can hide the selected molecular element. L is for label, which add the labels to highlight specific residues or atoms. So for example, if you know about the active site, then you can specifically highlight that residue, you can label that residue, because if you label the entire chain, then it will be very difficult to see that. So in this way, you can highlight the specific residue with the help of L. Then with the help of C, you can change the color scheme of selected molecules or region. So as we have seen that we can also type in the command, but if you do not want to type the command, then with the help of this C, you can change the color by selecting the specific chain or specific residue. And you can, if you want to change the color for the entire residue, then you do not have to perform the selection. You can simply click on C and change the color for the entire residue along with everything, along with ligand also. So now if we talk about S, which is show, so what it will show you, you can select the, for example in this case I have one HEW, so I can show it in different forms. I can show it in the lines, I can show it in the sticks form, I can show it in the form of spheres, I can show it in the form of cartoon, or I can show it in the form of ribbon. So it depends that how you want to see your molecule. So depending on your comfort, you can select any one of them, any one of them, but majorly the preferred one is the cartoons or sticks. So really these are the two different views in which you mostly see different protein structures in different publications. So now using the object panel to modify the representations. So for example, if you have opened it, it might open up in the stick format. So you want to change the representation. So how you can change? Either you can use the command. For example, in this case, firstly I need to enter the PDB ID and then keeping the function which is S in the comma and then greater than sign and then cartoon. So cartoon here tells you that in which representation you want to see it. So this is the cartoon representation. If you want to change it to stick or ribbon or spheres, then you can simply write that particular representation by this followed by the greater than. Then comes this H, which means hide. So this will hide lines. So this hides the default line model to improve the clarity. So now for example, firstly when I opened my structure, it was in the line format, but now I am showing it in the cartoon format. So what happened is that in that case, it do not hide these lines. It still shows lines also and cartoon also. So that makes it complicated. So what I can do is I can hide the lines. And why it is important? Because if I want to see spheres along with the lines or along with cartoons, so both at the same time I can see both the structures. So that is the advantage. So that's why we are provided with the help to hide function, so that we can hide the default line model which is opened, so that we only see the cartoon representation. Then we can also hide the waters, because the red crosses, they represents the water molecule. So we can simply write H greater than waters. So this will also hide all the water molecules which are present. So this is how we can use the object panel to modify the representation, that how we want to see our protein molecule or protein structure. So that if I want to publish it, then I can control it. I can show it is in the user's hand that how he want to visualize it or how he want to present the structure. Then default tabs in PyMOL for structural selection. So there are different default tabs. So one will be all, which will always be there. So it will select all the atoms including multiple PDB files if loaded. So in PDB you can also load multiple files. So in that case, all means all those files. So if you select, or if you click on all and if you click on color, so it will perform the function on all those files which are loaded. So that stands for all. Then in this case, one HEW, it selects all the atoms which are present in this particular PDB file. And if I select some residues or if I select the inhibitor, so then there will be another option or another tab which will open up. That will be my SELE tab, which means select tabs. So select tabs will select that will show you that what you have selected. So if you want to perform anything on that particular selected region, so that will be given in select. So in that case you can select that what action you want to perform on that region, or what do you want to show, whether you want to hide that region, whether you want to label those residues, or whether you want to change the color of those selected residues. So that is the third tab here. Now if we simply see this cartoon orientation, so we do not know what are the side chains, because we know that side chains of the residues are the ones which are taking part in interactions which vary. So if I want to know the main side chains or all those things, so how to view them? So we can choose the residue and we can type this command, which is set cartoon side chain helper, 1, or we can simply select the residue and then right click with the mouse, and then with the help of show option we can click on sticks. So in that way what will happen is that in my cartoon representation, those residues side chain will appear in the form of stick. So this is the stick representation. So this is how we can view the side chains. Now comes the measurement of distance between two residues. So if I want to calculate distance between any two residues, then what I can do is I can simply go to wizard, which is here. If we click on wizard, then it will open up multiple options from where we can select measurement, and after measurement we can click on distances. So there are different options available under measurement, such as distances, create new object, delete last object, delete all measurements, done. So now I can click on this distance, and now I have to select two residues. So after clicking on distance I need to select two residues. So in this case we can click on here and here, so in that way we can find the distance, or we can click on different residues. So depending on your choice you can find out the distance between any two atoms which are present in your protein structure, or elements which are present. And then there is this command also with which you can find it out. So set color name, then set color, and then you have to give the name. So generally the color which is preferred is the ruby red, which is generally preferred, so that it is easily visible. So in this case I haven't changed it to ruby red, this is the default color which they have shown. Then you can also save the image. So to save this image you can go to display, which will be in the menu bar, and then select background, and then we need to change the background color from black to white so that this structure is more clearly visible. So firstly we can select the white background, and then we can type this option in the terminal, which is ray 1080720, or we can simply go to this option which is in the right top side, which is the draw/ray. So from here we can decide that what width of the particular structure or particular image we want, what should be the height, or we can also decide the DPI, which tells us that whether the image which we will obtain, whether it will be of high resolution, or all those things can be decided with this. And then we can select two options, draw fast or ray slow. So this draw fast will quickly give you the image, whereas ray slow will take some time, but the image which it will provide you, that will be of better resolution. And with the resolution I mean to say that the cartoon representation which is given, so it will add some sense of depth and all those things, so that it becomes attractive and it becomes clear. So this is how, if we click on the draw fast and save it in the form of PNG, then this is how it will appear. So before this we have changed its background to white color so that it can be easily seen. And mostly in publications also, why we prefer white background, because this black background it will use a lot of ink, so to avoid the wastage also, that is also one of the reason. Now if you have a oligomer, so till now we have only seen the protein which only have a single chain, but what if we have a oligomer? So in PyMOL we can open our PDB file. So in oligomer we can change the color of different chains. So we can select the chains, because I have shown you that it have a sequence pane. So in this sequence, this will tell you that what are the different chains which are present. So by clicking on this sequence or selecting the sequence, and then select, or you can say SELE, that particular tab will open here. So with that you can change its color. And another thing which I have also told you, that in this sequence pane, from the display option you can also decide that whether it should give you the residue wise information or whether it should give you chain wise information. So with chain wise information, instead of selecting the entire residues in the chain, you can simply click on that one chain ID, 1 A B C, what is the chain ID, and then entire chain will get selected. So with the help of PyMOL, this is one of the advantage, that you can view the oligomer and you can change the respective color of each of the chain within that oligomer. So this is the another command, that right click on the particular protein molecule and then click on chain, then color by chain by chain. So these are two of options. So this you will find when you will click on this C. Okay, now you can also form a monomer from a multimer. So for example, if you have multiple such chains which are present, and sometimes what happens is that you have a homomeric protein in which all the chains are the same. For example, in case of hemoglobin. So in such scenarios, what you can do is that you can form a monomer from a multimer. So for that you need to open your PDB file and you can simply click on chain and then extract object. So in such scenarios, because each chain is referred to as different object, so in this way object 01 by default will be the selected chain that will be created as a different object. So in this way you are creating a monomer from a multimeric protein. And then you can also perform alignment. So as I have told you that in PyMOL you can open multiple PDB files, not just one PDB file. So that will appear in the right hand side. So in this I have written fetch 1 HEW. Now I can also write some another PDB ID. So that will also open another tab here, and both the protein structures will be loaded in this main window. So we can perform its alignment. For example, if there are two protein sequences which have similar structure and we want to align them, we want to see where they vary. Or in some scenarios what can happen is that you have different structures for the same protein, or if you have isoforms, and isoforms are the proteins which are obtained from the same gene. So in such scenarios those structures are highly similar to one another. So to see that where they vary, we can perform the alignments. So we can open both the PDB files, we can click on display sequence, and then we will view its name and chain numbers, and we can use this command which is align file name 1 chain A file name 2 chain A. So by putting this command we can simply see, we can align the different protein molecules. So this is one of the advantage of PyMOL, that you can also perform the alignment. Now we can also create a movie in PyMOL. So firstly we need to load the structure. So you have to go to PyMOL and you have to load your PDB structure. Now you can view it, you can zoom it, you can rotate it for your clarity, because in PyMOL you can simply click it, you can rotate it in either directions, you can zoom it. So all those things can be done. Then you have to set up the animations such as rotations, zoom, transition. You have to decide the key scenes. And for example, if I am moving it one side and then I am directly bringing it forward, zooming it, so I need to make sure that that animation is smooth. So stepwise we need to perform it. Then we will capture the keyframes, because we know what is the movie. Movie is simply different pictures which are moving with some time, which are moving at some speed, and then they form a movie like appearance. So similarly in this case also we will capture the keyframes, and those frames we will decide one by one that how each frame will be present. So we move to the starting point and save the key scenes. So one by one we will make rotations, zoom, transitions, and then we'll take their pictures. So they will be stored as the frames. They will be my key scenes. Now we can adjust the camera angles, we can zoom them, we can rotate them, and simply create a movie out of it. So for that we need to go to movie menu. Then we will select its animation type. We can also set the number of frames for smooth playback. So if frames will be high, then the playback will be quite smooth. Then we can preview it, we can adjust it, and then we can play with the help of play movie option. We can export this movie, and then this is how the movie appears. So this is also one of the fascinating feature of PyMOL, that you can create the movie. So this is kind of interactive view of the three-dimensional protein structure. And then after saving it in different formats such as MPEG, AVI or PNG frames, you can also adjust the quality and frame rate for the polished output. So this is how the movie can be created in PyMOL. Now this is the example in which how the PyMOL has generated such high definition images which are published in different papers. So this is from the PLOS 1 2012. So this is how it is represented. So in this case what they have done is that these are different regions or different residues which are represented in the stick format, and whereas the other residues or other protein molecule, it is shown in the form of the cartoon representation. So PyMOL tells you, or PyMOL provides you this ability that you can select which residues should be represented in which format, so that you can understand the interactions taking place at the active site, or how my protein is interacting with the ligand. So all those things becomes clear by using the PyMOL as a molecular visualization tool. Similarly, in this case also, some residues are shown in the sphere form, whereas others in the cartoon form for easy representation. In this case also, they have shown the electron density map for the selected region, and then these are some of the residues. And in this case also, different sites and how they are interacting with different tryptophan moieties, and these are different structures, different residues which are represented in the form of stick, and this is the ribbon representation which is in the back side. So this shows you that how, with the help of PyMOL, you can decide that in which representation you want to see the structure. So these representations make it easy to see how the things are happening. Now we'll talk about the WinCoot and PyMOL together, that we can also mutate the residues. So instead of performing the mutations, taking long time to analyze the structure by using X-ray crystallography and all those things, these PyMOL and WinCoot, it also provide us advantage that you can simply mutate the residue and you can see how the structure can change by mutating the residues. So in WinCoot, we can make the mutants. So for that, you need to open the PDB file in WinCoot. You need to choose draw, and then you have to go to atom option. So this will then you have to select the residue which you want to mutate. So it will open up a drop down menu from which you can select the chain. For example, if your protein has multiple chains, so in that case you can select the chain, then you can select that residue, and you can also select the residue to which you want to mutate. Then you have to click on apply, then click simple mutate on the right hand menu and click on the residue. So in this case, you are firstly selecting which residue you want to mutate, and then you are going to simple mutate, and then in that simple mutate option you are selecting the new amino acid residue which you want to change it with, and then you can select the mutated file, you can save its coordinates, and then you can open that file with PyMOL. So open the wild type and mutated PDB files. So in this case, wild type will be the one which contains the previous residue, whereas the mutated will be the residue which we have changed. So we can align these two structures, keeping the wild type as a template, so that we can identify where the mutated residue is present and how it has changed. So for example, in this case, previously there was glycine, now we have mutated it, previously it was glutamine, and now we have mutated it to lysine. So we can see how the structure has changed. So all those things can be done with the PyMOL. After making this mutation in WinCoot, we can open it with PyMOL and we can see that how it changes. So for easy representation, for that particular residue, we have selected that it should be shown in the form of stick. And in other residues, all the residues in the protein, they are still shown in the cartoon representation. So because we can simply distinguish among them, so that's why we have selected it in the form of stick. And this is the change. So now we can see how the structure is changing by mutating this residue, because we know that this glutamine and this lysine is positively charged. So in this case, this shows you that with the change in the residue, it can also make new interactions. So all those analysis can be performed later on. So after doing all these things, we can colored by the elements. We can choose the color. For example, oxygen can be represented with red color and all those things. So we can decide that which color we want to show, so that we can easily figure out that what that residue is. We can also annotate it, we can label it. So in this case, they have labeled that what is the residue, what is their position, and then we can render the image. So we can unselect the mutated PDB, we can perform the ray trace for wild type and save the image. Then we can unselect this wild type and then select mutated PDB and perform ray trace and save. So in this way, we can also save the session, so that in future also we can use it. And ultimately, we can use the PowerPoint to label this or annotate this, because we have these two figures separately. So this is done with the help of PowerPoint, that we have specifically shown that this is the region and here is the cropped image. So all these things, all these visualizations, all these representations, So they are facilitated by the PowerPoint so that they become visually attractive and easy to understand. Now we will move to practice questions, and in the end, if we have some time, then we will look how the PDB looks like, how we can perform different search operations, and what are different file formats which appears. So now let's begin with different practice questions. So the first question is, which of the following is incorrect about the Swiss PDB viewer? Option A, it is a structure viewer for multiple platforms. Option B, it is a structure viewer for single platform. Option C, it is capable of structure visualization, analysis and homology modeling. And option D is none of the above. So you have to tell that what is incorrect about Swiss PDB viewer. So I'll give you some time to think and then we'll discuss the answer. Okay, then let's see what is the right answer. So the right option is option B, which is it is a structure viewer for single platform. So this option is incorrect. Now let's one by one see each option. So first option was that it is a structure viewer for multiple platforms. So this was correct because Swiss PDB viewer is available for multiple operating systems, such as you can open it in Windows. It also supports Linux operating system. It also supports Mac OS. So that means it is a viewer for different platforms, multiple platforms, and not just a single platform. And then third option was that it is capable of structure visualization, analysis and homology modeling. So this is correct. We can perform all those things. Now let's move to next question. So next question is, which command or tool is used for high definition images in PyMOL? Option A, deselect. Option B, ray. Option C, zoom. And option D is rock. Okay, so let's see what is the right answer. So right answer is option B, which is ray. We have just seen that with ray we can decide that what should be the resolution. We can select DPI and all those things so that the images which appears, they are of better definition, high definition, and they are visually attractive. So it generates high resolution ray traced images in PyMOL. Whereas if we talk about deselect, so it removes the current selection. So in selection, if you have selected some chain or if you have selected some protein, so with the help of deselect, you can deselect it. For zoom, this is just to adjust the view. You can zoom the particular structure. Then if you talk about rock, then it animates the structure by oscillating it, but it does not affect the image quality. So this is how different ray trace modes exist. So this one is the default mode, which is set ray trace mode to 0, which is the default rendering mode for PyMOL, displaying natural colors. So these are natural colors. If we talk about trace mode 1, so this gives the natural colors with black outline. So you can see in this case these are the natural colors, but here you also find that there are some black outlines which are there now. So you can simply see from here. So this is looking more attractive or more clear, more high definition. If we talk about ray trace color green set ray trace color mode 3 ray, so in this case we can also decide that whether the outline should be black, green, red, depending on our choice. So in this case this outline is of green color. So this is how its representation can change. So in this way, this is to show you that how with the help of ray you can generate the high resolution images. And this is also one of the example. So in this case this is the non-ray traced image, and the right one is the same scene, but in this case we have performed the ray tracing. So you can see how clearly it appears, how high definition it appears. Now let's move to next question. So the next question is, which of the following is present in the model fit refinement tool of COOT? Option A, Ramachandran plot. Option B, simple mutate. Option C, environmental distances. And option D is go to atom. So you have to tell that which of the following is present in this tool of COOT. Okay, now let's see what is the right answer. So the right answer is option B, which is simple mutate. So if we talk about option A, so Ramachandran plot, it is although present in COOT, but it is mainly for the validation and not for fitting and refinement. Simple mutate, it is correct because it allows the direct mutation of residues in the model. So in that way, it is the part of model fit and refinement tools. And we have also seen that by selecting the residue and going to simple mutate, we can decide that to which residue we want to mutate our structure. Then C was environmental distances. So these are present, but it is more of the analysis tool rather than the model fit and refinement tool. And if we talk about go to atom, then this is the navigation tool in which we are just selecting that particular residue which we want to mutate. So we are just selecting here, so it is a navigation tool and not used for refinement. Okay, now let's see our next question. So what is the important software to make the diagrams? Option A, PyMOL. Option B, Microsoft PowerPoint. Option C, WinCoot. And option D is all of the above. So you have to tell which one is the important software to make diagrams. Okay, so now let's see the right answer. So the right answer is all of these. So we have already seen PyMOL, WinCoot, and we have also discussed that Microsoft PowerPoint is also very important to make the diagrams. So that's why option D is correct. Now let's move to next question. So next question is, what is the command to view the side chain of a residue in PyMOL? Option A, set cartoon sidechain helper, 0. Set cartoon sidechain helper 1. Set cartoon sidechain helper, 1. SELE, that means select. And option D is none of the above. So now you have to tell which command will help you to view the sidechain residues. Okay, now let's see the right answer. So right answer is option B, that is set cartoon sidechain helper 1. So when we say 0 after any command, and these commands they all means all the letters will be in the lower case, that is small letters. So whenever we say anything followed by 0, it means we are disabling it. So it disables the sidechain display in the cartoon mode. And then if we keep 1, that means it will enable the display of sidechains for polar charged residues, for example arginine, lysine, glutamate, aspartate, histidine when in the cartoon mode. And then if we talk about 1 SELE, so this is the cartoon sidechain helper setting, but it does not take a selection argument. So this is also not the right answer. It does not take selection into consideration. And option D is also incorrect because our right answer is option B. Now let's move to next question. So the question is, which of the following parameters cannot be obtained from the validation report from WWPDB? Option A, entry composition. Option B, data and refine statistics. Option C, resolution and model quality. Option D, macromolecule fold. Okay, now let's see the right answer. So the right answer is option D, which is the macromolecule fold. So with the help of validation report, we can get the information about the entry composition, which will tell you about the details of the molecule, that is protein molecule, its chains, and if it is if it is complexed with the ligand, then that ligand information will also be given. Second is data and refined statistics. So this refinement details are also present, such as R factors and B factors. Then a resolution and model quality, so it is also present. It tells you about the resolution, clash scores, geometry validations. So all three properties, they are present. But if we talk about the macromolecule fold, then it is not provided in this WWPDB validation report, and these are generally analyzed separately using scope or cath. Now this is how the full WWPDB file looks like. So just give me a moment. Yeah. So there is some network issue on my end. So this is how the WWPDB X-ray structure validation report files appears. Okay, just this is better. This is even better. Yeah, so now this is how the validation report appears. Firstly, it tells you about the different details about that particular molecule that for which that report is present. So in this case, it is for one ORF. This is the title, and these are the authors who have submitted it. This is the date at which it is deposited, and this is the resolution which it will tell you about the structure. These are different parameters, different informations which are given, such as overall quality, entry composition, residue property plots, data and refinement statistics, model quality, two close contacts, torsion angles, non-standard residues and ligands, polymer linkage issues, fit of the model and data. So in this way, these all things are mentioned in the validation report. And in the end, you can see that whether the protein structure which is present, whether it is in the favorable region, and what are the different things. So this is about the validation report. Now let's move to next question. So our next question is, now we can see here, so which is the energy minimization of a model protein can be done using option A ChemSketch, option B MolDraw, option C RasMol, and option D is Swiss PDB viewer. So you have to tell that which can be used for the energy minimization of a model protein. Okay, so the right answer is option D, which is Swiss PDB viewer. So if we talk about ChemSketch, then it is used for drawing the chemical structures and it is not used for the protein energy minimizations. Whereas if we talk about MolDraw, then it is used for the molecular drawings and not for the energy minimizations. If we talk about RasMol, then it is also used for the molecular visualizations. Whereas this Swiss PDB viewer, it is used for the energy minimizations for refining the protein structures. Yeah, so next question is, what is the format for command of RGB color in PyMOL? And these are the different options, so you have to tell which one is the right format. So this also we have discussed. So the right answer is our option C, in which these three values are written in square brackets, as we have seen during this PyMOL session, that how we can select the color. So these values corresponds to R, G and B values. Okay, so our next question is, which one of the following is a command based offline tool for molecular structural visualizations? Option A Swiss PDB viewer, option B RasMol, option C QMOL, and option D is PyMOL. So you have to tell which of the following is a command based offline tool. Okay, now let's see the right answer. So the right answer is RasMol, which is a command based offline tool for molecular visualizations. The Swiss PDB viewer, it is GUI based, that is graphical user interface, and it is not primarily command based. So primarily we do not have to enter the commands, simply we can use the GUI interface. If we talk about QMOL, then it is GUI based, completely GUI based, and there is no such command line thing. If we talk about PyMOL, then it has command line capabilities, but primarily it is GUI driven. We have seen that there are certain options so that are available. So RasMol is command line based. Now our next question is, which of the following is an incorrect statement? Option A, WebMol is a web-based program built based on a modified RasMol code and thus shares many similarities with RasMol. Option B, WebMol is a web-based program that is totally different from RasMol. Option C, KIME is a plugin for web browsers. Option D, KIME is not a standalone program and has to be involved in a web browser. Okay, now let's see the right answer. So the right answer is option B, which tells you that which one is the incorrect statement. So when we talk about first statement that WebMOL is a web-based program, so that is true. It is a web-based program and it is also based on modified RASMOL code. That's why it shares many similarities with RASMOL, so this is true. Whereas second option was that it is a web-based program and totally different from RASMOL. So this is incorrect because it is based on the modified RASMOL. Then option C was that KIME is a plugin for the web browser, so this is also true. And then next option was KIME is not a standalone program and has to be involved in a web browser, so this is also true. Yeah, so now this is the difference between the RASMOL and the WebMOL. So in RASMOL, this is entirely based on the command line. So it is a simple molecular viewing program with limited manipulations. Freeware, that's why it was considered the granddaddy of visual tools. It is a standalone program and source code is also freely available. It is run on multiple platforms such as Mac, Windows, Linux, SGI and Unix. And this is the URL. Whereas this WebMOL, which is based on the modified RASMOL, so this is a Java applet and downloadable application. So you can download it. There are different tools in WebMOL such as distance, angle, dihedral angle measurements, steric conflict detection, interactive Ramachandran plot, distance plot. So these are different tools due to which it is more interactive and more manipulations can be done, which were limited in the case of RASMOL. And this is also compatible with Java browsers such as Internet Explorer, Safari, Mozilla Firefox. So means it is also supported by multiple platforms. This is a WebMOL, which is online tool you can say, and you can also download this application as well. So this is the difference between the WebMOL and RASMOL. If you are interested, then you can use them, you can go through them. Now the next question is, which scientist is credited with developing the first molecular model using the ball and stick representations? Option A, Linus Pauling. Option B, Francis Crick. Option C, John Kenju. And option D is Max Perutz. Okay, now let's see. So the right answer is Linus Pauling. So you might think that why not John Kenju or Max Perutz, because they also gave. So before them, Linus Pauling was the one who gave the first molecular models using the ball and stick representation. Now next question is, which file format is most commonly used to store the atomic coordinates of macromolecular structures? Option A, PDB, option B, MMSIF, option C, MTZ, and option D is FASTA. Okay, this is quite clear. So the right answer is option A, .pdb, because in this format, as you have seen, it provides you the information of x, y and z coordinates, which are the atomic coordinates at the level of atoms. Whereas if we talk about MMSIF, then this is a macromolecular crystallographic information format in which it tells you about the unit cell geometry, ABC, alpha, beta, gamma, those angles. So it tells you about the crystallographic information and not the atomic coordinates. Similarly, MTZ, this tells you about the electron density map. .FASTA, it only provides you the sequence based information and nothing to do with the structure. So that's why our option A is correct. Now our next question is, what is COOT primarily used for in structural biology? Option A, molecular dynamics simulations. Option B, protein sequence alignment. Option C, model building and refinement of macromolecules. And option D is ligand docking and interaction studies. Okay, now let's see. So the right answer is option C, which is model building and refinement of macromolecules. So we have seen that we can perform the refinements, we can use different constraints, Ramachandran constraints or all those things we can decide, and we can refine the structures. Along with this, we can also perform mutations and we can see that how the mutations bring about change in the structures. So in this way, these can be done in the COOT and not protein alignment or molecular dynamics or ligand docking. So these things are not done. Now let's move to next question. So which command in PyMOL is used to color different chains in a structure? Option A, color chain. Option B, spectrum count chain. Option C, set color chain. And option D is color by chain. So you have to tell which command in PyMOL can color the different chains. Because sometimes what can happen is that in PyMOL you have a protein in which there are multiple chains which are present. So you want to see them differently. Okay, now let's see the right answer. So the right answer is option B, which is the spectrum count chain. So this is the command which you use if you want to change, if you want to color different chains separately. So in all these options, you have not mentioned that you want to change color for each chain. These are not the right options. So option B was right, spectrum count chain. Next question is, which of the following features is not available in PyMOL? Option A, surface representation of molecules. Option B, protein structure modeling from sequence. Option C, ligand protein interaction visualization. And option D is ray traced image rendering. So you have to tell which is not available in PyMOL. Okay, then let's see the right answer. So the right answer is option B, that is protein structure modeling from sequence. Because we have seen we can represent the molecule surface, we can also study protein ligand interactions, and we can also get the ray traced images in PyMOL, but we cannot do the protein structure modeling from a sequence. It is not possible that you provide the FASTA file and get the structure. So that's why option B is the right answer for question 15th. Next question is, which metric is used in structure validation to evaluate the steric clashes in macromolecules? Option A, clash score. Option B, Ramachandran plot. Option C, R free. And option D is electron density map. So the right answer is option A, that is clash score. So this is for the steric clashes. And this is how the validation report appears. Its first column, its first type is overall quality at a glance, in which it gives you the information of clash score, Ramachandran outlier, side chain. So these are the different informations which will be present in the structure validation report. So our next question is, which molecule is not typically found in a PDB file entry? Option A, protein. Option B, water molecules. Option C, DNA, RNA. And option D is ATP energy levels. Okay, so this question is also quite straightforward. So the right answer is option D, ATP energy levels, because in PDB file protein is for sure, water molecule is for sure, because proteins interact with water molecules. So water molecules are generally used to make the protein in the aqueous environment, so they will always be there. If we talk about DNA, RNA, so in many scenarios proteins are complexed with other small molecules. So in such scenarios there could be some RNAs, sugar, DNA. So all these things can be seen in the PDB file entry, but ATP energy levels, they cannot be found. They are not typically found in the PDB file entry. Next question is, which refinement program is commonly used for crystallographic model refinement? Option A, Autodoc. Option B, Phoenix. Option C, Blast. And option D is Chimera. So now you have to tell which refinement program is commonly used. So I think you have not gone through this in the class. So now let's see. So the right answer is option B, Phoenix. And this is quite important. That's why I've kept this question. So Phoenix, this is the tool this is the program which is generally used nowadays for the crystallographic model refinement. This is how its homepage looks like, and this is the publication from where I have taken it. So this is highly annotated software system for macromolecular structure determination, and it provides an initial partial model of structure without significant human interventions, given moderate resolution and good quality data. So because of new algorithms for maximum likelihood molecular replacement, heavy atom search, template and pattern based automated model building, and automated macromolecular refinement. So these algorithms, they are based on comprehensive set of crystallographic libraries which are available to the community, and they are easily accessible to users through the Phoenix GUI and command line. So this shows you that this particular refinement tool is generally used for the crystallographic model refinement. Now the next question is, what does a z-score greater than 5 indicates in bond length or angle validation? Option A, the structure is perfectly modeled. Option B, a significant deviation from ideal geometry. Option C, there are no errors in the model. And option D, the structure is at atomic resolution. Okay, so this also I have kept because I want you to understand that what a validation report means, how to study it, how to read it. So for this question, the right answer is option B. So if the z-score is greater than 5, then it means that there is a significant deviation from the ideal geometry. So when we were talking about the validation report, then we have seen in that example that in some times the geometry is ideal, but it is not favoring some other metric, due to which there were some red dots which appeared although the color was green. So in this way, z-scores, it tells you about the geometry, which takes into consideration the bond length and the bond angle. So this z-score for a bond length is the number of standard deviations the observed value is removed from the expected value. So when we compare among observed and expected values, so this tells you about that how many number of standard deviation the observed value is. So bond length or angle with z value greater than 5 is considered an outlier worth inspection. That means you have to inspect it. And RMSZ, which is the root mean square of all the z-scores of the bond lengths or angles, so that is RMSZ, and this is the z-score. So now this RMSZ also has some guidelines which you need to make sure. So the general guideline for the ideal geometry is that the bond lengths, so for bond length, RMSD value should be approximately equals to 0.02 to 1. Ideally it should be less than 1. If we talk about bond angles, then for them RMSD value should be between 0.5 to 1.5, but ideally it should be less than 2. So higher RMSD value, which is greater than 2, it indicates that potential modeling error or poor refinement, whereas lower RMSD value, that is less than 0.5, it indicates that there is over restrained refinement. So the balance should be there, that the RMSD value should be greater than 0.5, but it should be less than 2. So in this case, if we talk about RMSD, then it is 0.6. That means greater than 0.5. And in this case, for bond angle, it is 0.87. Then in this case also it is less than 2 and greater than 0.5. So that means my particular structure for this particular protein, which is 1-ORF, this is having the ideal geometry. So this is about the z-score. Now let's move to our next question, or the last question, which is what percentage of residues in a well-refined protein structure should ideally be favored in the Ramachandran region? So option A is 50%, less than 50%, option B 50 to 70%, option C 70 to 85%, and option D greater than 90%. So for well-refined protein structure, what should be the ideal percentage? So this is also kind of straightforward only. So the right answer is greater than 90%. So if the percentage of residues which are favored are more than 90%, then that means your protein structure is well refined. So with this, I will end today's session. And I hope you got all the concepts which we have discussed so far. And by looking at the time, I think we do not have enough time to go through PDB, but once I'll give you a glimpse that how we perform search operations in PDB. So first of all, we need to enter PDB in our Google search engine. Then we can go to this RCSP PDB homepage. Now in this search option, we can either enter a PDB ID, or if you do not remember the PDB ID, then you can simply enter the name or anything like kinases, transferases, any name you can enter. So for now, I know one of the ID, which is 1HEW, which we have studied in our example. So by entering this ID, I can click on this. Now it will open up this entry. Now from this display file, we can see that in which format we want to display it. So there is a FASTA sequence format, MMSIF format, and then PDB format. So these are different formats. We can also download it either in the FASTA sequence. So these are different download options which are available in PDB. This is the validation report. So by clicking on full report, you will be taken to the page in which the full report will be given. So this is how the report appears. These are different metrics. You can see different values, clash values. You can see entry composition. So these are the things. Now coming back to display file. So if I click on FASTA sequence, then it will give me this representation in which this is the FASTA sequence. So due to certain issues, yeah, I hope now it is visible. So this is how the FASTA file generally appears, starting with the greater than symbol, and then this is the comment, and this is the sequence which it appears. Now I want to display it in my mmsif format, for example. So this is the mmsif format. So I told you that how it is different from SIF, that it gives you all these information as well: structural model information, citation information, journal information. All these informations are given. If we scroll further down, because the SIF files is for the crystallographic information, so if we scroll down, then we'll see the information about the crystal, which is X-ray diffraction information. So this tells you about the ID, which method has been used, and these are different informations. All these informations can be found here, and this also gives you the p bond angle and all those things. Let's scroll further down to get more information. So structure, configuration, there are multiple things which are given in these files. So it depends that what information we need. So because they contain a detailed information, sometimes some information we do not know whether it is important or not. So most of the residues are given. Their coordinate information. So in this way, this mmsif file, it contains the entire information about my macromolecules. Then these water molecules, which are heteroatoms. So in this way, this is my mmsif file, which tells me about the crystallographic information and all the details about the macromolecular protein, and its validation report as well as, and different connections between different residues. All those things are given in this. So we can simply say that it is a detailed information. And in some places, as you can see, there is a question mark. So that means if certain things are not identifiable, then they are given in the form question mark. And why they are given the question mark and not changing it? Because it will disturb the format. So that's why even if some informations are not available, then they are still kept. So this is about my mmsif file. Now let's see the another format quickly, which one is my PDB format. So this is the PDB format, starting with header, then title, compound, and all the informations which is relevant, remarks. So in remarks, different things, isotopic thermal restraints, all the information will be given in the remark, which is about the analysis, validations, and all those things. Then when from here it starts, from here the atomic information is given. So this atom tells you atom wise x, y, and z coordinates. So these are x, y, z coordinates. Then B factor and occupancy. Sorry, this one represent occupancy, and this 30.48 is the B factor, and this one is the atom or the element. So atom 1 means N, that means nitrogen of lysine, which is present in A chain. 1 means residue. So lysine is my first residue. And this is my x coordinate, 9.981 my y coordinate, and 10.408 my z coordinate. This is occupancy 1, which means it is always present in that particular position only, that particular conformation only, and 30.48 is my B factor. So in this way, you can scroll down and you will be able to see that what are the different atoms and different residues which are present. So this one is the atom count, and this one is the residue count. This one is the chain number. So now let's scroll down till we find the end. So as soon as my atoms end, there will be a ter record, which will tell me that this is my terminal residue, which is my leucine. So this is leucine is present towards the C terminal end. Then comes heteroatom, which will tell me that what are the other atoms other than my protein. So in this case, it has an inhibitor NAG in my this particular file, and then heteroatom starts. So this NAG, after this NAG ends, there is water. So water is also heteroatom. So then water is present in the chain A, and ultimately this heteroatoms will end. Then there will be a connect record. Connect record will tell you that where are the macromolecular interactions, which of the residues are interacting. So that information is given, and this is the different other interactions, dihedral angles, and all those informations are given by connect. And ultimately there will be a master, which will give me all the information about different index values of different things, and then ultimately followed by end. So this end specifies that my PDB file is ending here. So these are different display formats, and you can also download them. On the left hand side, you can see this is the representation. You can explore it in 3D. And if you download it in PDB format, you can open it in PyMOL. And then you can see, if you scroll down, there are multiple details which are added. From UniProt, you can look for the sequence. Yeah, so I won't go into the detail again because it will take a lot of time. Yeah, so this is about today's session. So we'll see you in the next session. And if you have any doubts, then you can ask it in the discussion forums. So I'll be happy to answer your doubts. And sorry for today's network issues. So thank you so much.